So for those who are unfamiliar with the research, let's go through some key points and get some more clarity, shall we? So, James began his parade of dishonesty right at the beginning of his opening statement by attempting to reframe the topic of debate in a dishonest way. He knows that the idea that we are clueless about the origin of life is totally indefensible.
Now you may say that there are alternative ways of building up nucleotides, and perhaps there was some geochemical way on the early Earth. But what we know of the experimental difficulties in nucleotide synthesis speaks strongly against any such supposition. However, it is to be put together, a nucleotide is too complex and metastable a molecule for there to be any reason to expect an easy synthesis.
On to the opening statements, and what would a James Tour opening statement be without some quote mining? Let's start with what he says about Jack Szostak.
Professor Jack Szostak, then at Harvard, now at the University of Chicago, said in 2014 that he'd create life in his lab in 3 to 5 years. He did not.
Jack claimed in 2014 that he could get a protocell in 3 to 5 years. Did it happen? No, so we are clueless. Well, let's see what he meant in this Huffington Post article. Oh, he meant that given the building blocks in abundance, it could be done, because Jack was specifically referring to protocell assembly, and not the synthetic chemistry that other members of the community are working on.
That is the qualitative difference between actual cells and lifeless vesicles, basically water bubbles, okay? So here's E. coli, a photomicrograph. There's a world of difference qualitatively between that thing and a simple lipid vesicle. So Deamer, thinking about this, about that qualitative difference, he says, let's imagine the following experiment. He says, I will grant you, my colleagues, my other origin of life researchers, I will grant you all the molecular hardware of a normal bacterial cell. How do we do that? What we do is we start with a cell, call it a Humpty cell, and we hit it with sound. So you can sterilize a bacterial culture with sonication. Sound at the right frequency disrupts the membranes of cells. So we have one of these cells alive in a controlled setting where we disrupt it by sonication, and now we ask what's going to happen. So Deamer describes this, and he says, we'll take the contents of that cell whose membrane we have disrupted, and we'll put it in a flask with nutrient broth. So we'll give it all the food that it could possibly want. What do we have there? What's the hardware present in our flask? Well, we have entire bacterial genomes. All the DNA is there. The dream of a bottom-up researcher, to have all the DNA specifications present, together with ribosomes, the big and most critical molecular machine that turns nucleic acid into proteins. We've got whole ribosomes, complete genomes. He goes on. Membranous vesicles, ATP, the gasoline, if you will, the petrol, the main energy source in all life is present. Thousands of enzymes, right? Proteins that will do particular tasks for you. In other words, you have, under the Humpty Dumpty experiment, all the parts are present. All you've done is you've disrupted the membrane of the cell, but all the hardware is there. Now, what's going to happen, Deamer asks? Nothing. What will happen is water will begin to attack these molecules. It will attack the peptide bonds in the proteins. It will attack the nucleic acids. What will happen is little, Deamer goes on, little would happen other than the slow degradative reactions of hydrolysis. So water is going to start doing what water does chemically, and the bits and pieces, the hardware, the molecular constituents of life will begin to break down. The first thing that you will lose probably will be the ATP, all right? But pretty much everything's going to be lost eventually. And Deamer finishes his description by saying, you've got the entire complement of molecules associated with the living state. What's gone? What's gone is the relationship between those parts. What I described in the thought experiment on planet Z beta, that relationship of those functions has been lost.
He quote mines him again to pretend that autocatalytic sets don't exist.
Jack Sostek now says that aside from nature's construct of RNA, invoking of, quote, autocatalytic sets was never chemically realistic, unquote. Hence, the prebiotic relevance of autocatalytic sets crumbles.
Wow, I guess all of these papers and reviews on autocatalytic sets aren't real because Jack said so. But did he? From the same article. If you read, you immediately see that he was talking about something very specific and the semantics of how this term is defined. When origin of life researchers disagree about the nuance surrounding hypotheses within their field, James pretends they are saying origin of life research is nonsense. Another instance of James trying to hand-wave away an entire field of science he doesn't like, while having the audacity to pretend that it's a member of that field who's doing the hand-waving.
Ribozymes constituted a shocking and promising discovery. There was now genetic material that was catalytic as well. Many found the metaphor appealing, a world with a jack-of-all-trades RNA molecule, catalyzing the formation of indispensable cellular scaffolds, from which somehow then cells emerged. Others were quick to notice several difficulties with that scenario. These included the lack of templates enabling the polymerization of RNA in the prebiotic complex mixture and RNA's extreme lability at moderate to high temperatures and susceptibility to base-catalyzed hydrolysis.
All James had to do was search the literature, and he didn't do it. Continuing, James tries to extrapolate this toward pretending that no one has made RNA at all, regardless of linkage.
Without an enzyme, it has never
You
been
are completely clueless.
done. It's
This
over.
has been done.
It's over. You can't make RNA, there's no life. You can't make RNA, there is no life.
As though heterogeneity of linkage makes the RNA somehow not even qualify as RNA. So I again refer to the absolute common knowledge that RNA has been formed prebiotically for decades.
There are many weak points in this hypothesis. For example, 1. Nucleotides have not been produced from simple inorganic compounds through prebiotic means and have not been detected in any meteorites, although a small quantity of nucleobases can be obtained. 4. 2. It is quite difficult or most likely impossible to synthesize nucleotides and RNA through prebiotic means. 3. It must also be impossible to self-replicate RNA with catalytic activity on the same RNA molecule. 4. It would be impossible to explain the formation process of genetic information according to the RNA world hypothesis, because the information is comprised of triplet codon sequence, which would never be stochastically produced by joining of mononucleotides one by one. 5. The formation process of the first genetic code cannot be explained by the hypothesis either, because a genetic code composed of around 60 codons must be prepared to synthesize proteins from the beginning. 6. It is also impossible to transfer catalytic activity from a folded RNA ribozyme to a protein with a tertiary structure.
Been showing him. So here he is silently conceding something that he fiercely denied over 2 years of content because he knows it's indefensible.
You're going to do condensation reactions in water? I don't understand why some would suggest that you do condensation reactions in an ocean like this, in water. How can that be? Tell me about that. How do you do condensation reactions in water? Have you any references on these claims?
The papers I brought for this topic demonstrate what I had been saying in my content, that chemical activators make this chemistry possible, and there are plenty of prebiotically relevant ones, which have been verified experimentally, which he would have learned 2 years ago if he had actually bothered to check the literature. But James knows he's absolutely screwed here, so he has to pretend that he never said anything like that, which he does by immediately shifting the goalposts and complaining about potential reactions from side chains that would interfere with peptide formation.
To start polymerization, the amino acids would have to be concentrated all together at one assembly site. That would be a huge challenge in the ocean. In an aqueous environment like an ocean, the concentration of reactants can be diluted due to the large volume of water present. Lower reactant concentrations make it more difficult for condensation reactions to occur efficiently. There are 4 different ways to bond amino acids together by the side chains. If bonded to the wrong side chain, the product would be meaningless. To form a chain, it is necessary to react bifunctional monomers, that is, molecules with 2 functional groups so they combine with 2 others. If a unifunctional monomer, with only 1 functional group, reacts with the end of the chain can grow no further at this end. If only a small fraction of unifunctional molecules were present, long polymers could not form. But all prebiotic simulation experiments produce at least 3 times more unifunctional molecules than bifunctional molecules. The average size of a protein 300 to 400 amino acids. To get a polymer chain from a mix of bifunctional and unifunctional monomers where the predominance is unifunctional monomers is in the realm of the impossible. While condensation reactions can lead to the formation of larger organic molecules, the stability of these molecules in water needs to be considered. Water can also catalyze hydrolysis reactions, which are the reverse of condensation reactions. Hydrolysis breaks down larger molecules into smaller ones by using water molecules to split chemical bonds. In a water-rich environment, the presence of hydrolysis can compete with and potentially hinder the formation and stability of complex molecules. The hydrolysis of peptide bonds results in the cleavage of the chain, breaking it into smaller peptide fragments or individual amino acids.
Well, for some more insight, I asked Dave Deamer for a comment, which he provided in the form of a note briefly summarizing some facts about this field, in which he is one of the world's top researchers. He goes all the way back to the 1980s to discuss the first RNA synthesis using a template but without enzymes, then over to 2006 and the first RNA synthesis without any template but using catalytic clay surfaces, then to some of Shostak's work regarding protocells, and then Deamer's own work involving polymerization of monomers without activation via wet-dry cycling in prebiotic analog conditions at Yellowstone. RNA made without activation, without human intervention, in a hot spring.
The Deamer hypothesis proposes that the polymerization of RNA could have occurred in hot springs, such as those found in Yellowstone National Park. There are several challenges and limitations associated with this hypothesis. Hot springs are characterized by high temperatures, which can be detrimental to the stability of RNA molecules. RNA is susceptible to degradation at elevated temperatures, and the RNA backbone can undergo hydrolysis, leading to the breakdown of the molecule. The ability of RNA to withstand and maintain its structural integrity in the extreme heat of hot springs is a significant challenge for the Deamer hypothesis. In hot spring environments, the high temperatures can promote reverse polymerization reactions, where RNA strands break down into individual nucleotides. This reverse reaction hampers the accumulation and maintenance of longer RNA chains necessary for the development of functional RNA molecules. Hot springs often have high water flow rates, resulting in dilute solutions. The low concentrations of necessary reactants, such as nucleotides, in such environments can impede the efficient polymerization of RNA molecules. Adequate concentrations of nucleotides are required for the successful formation and elongation of RNA chains. Hot springs contain various minerals and metal ions that can interact with nucleotides and RNA molecules, potentially inhibiting or interfering with their polymerization processes. These chemical interactions may lead to the formation of stable complexes or hinder the stability and functionality of RNA. While the Deamer hypothesis provides a mechanism for the emergence of RNA in hot springs, direct experimental evidence supporting this specific scenario is limited. Conducting experiments in hot spring-like conditions to demonstrate the polymerization of RNA and its stability under such extreme conditions is challenging.
Maybe for some additional clarity, we can consult another expert here. How about Lauren Dean Williams, an origin of life researcher over at Georgia Tech? What does he have to say about where we're at in terms of prebiotically plausible synthetic pathways to these molecules? What is your view in terms of the status of the field with prebiotically plausible synthetic pathways to get to, say, polypeptides and polynucleotides?
Okay, those are two different stories. Polypeptides, it's a pretty clean story. You know, you can make amino acids very easily by prebiotic methods. They're raining down on our heads from outer space, and we now have this wet-dry chemistry to polymerize them, well, oligomerize them. So there's very plausible prebiotic mechanisms that are very easy to do to get to, you know, they're not proteins or anything like that, but they are short. Oligomers are short, and they are not exactly clean. They have some mixture of peptide and ester and some other things, but it's amazingly close considering the very simple, easy chemistry you can do to get to them. You know, we don't know where RNA came from. Maybe it came from direct chemical synthesis on the ancient Earth, or maybe it's a product of chemical evolution. Meaning, you know, and an evolution can make new things. We think chemical evolution can make molecules that were not there originally on sort of the ancient prebiotic Earth.
You mean like something else was produced, like short oligopeptides that had catalytic activity?
Yes, we think catalysis is part of it. You know, one of the things that we think is very important is actually sort of selection on the basis of persistence, we call things that live longer win. Even in biology, this is true. But so the idea is that peptides are great at assembling. They, you know, that's what a beta sheet is, et cetera. They're assemblies are resistant to degradation. So if you can just by any random process make molecules that assemble, then their population will rise because they degrade very, very slowly. This is true. It doesn't matter whether it's RNA or DNA or protein or any kind of molecule. Once you assemble it, it becomes resistant to degradation. So we think in early chemical evolution, selection was really on the basis of protection from degradation.
Mm-hmm. So it's just basic statistical dynamics, essentially. What about something I've tried to learn about is even further protection from degradation by interaction with mineral surfaces or things like that?
Yes, you know, any kind of assembly. In fact, if you just take anything and crystallize it, that's a self-assembly, right? Crystals are, you know, you can irradiate them, you can do things, and they're just chemically tougher than the naked molecules out in solution. So and it can be a self-assembly or assembly with another molecule or on a mineral, anything. Any kind of assembled molecule is resistant to chemical assault, is one of the ways to say it. And there's so many ways that things can assemble.
Right. So it sounds like there's some consensus as to that clearly we need polypeptides and we need polynucleotides, but we're not really quite sure how it went incrementally in terms of which appeared first and helped catalyze the other. But all of this is to say that we're far from clueless.
We're not clueless. No, we are not at all clueless.
An enormous amount of empirical data have established, as a rule, that organic systems, given energy and left to themselves, devolve to give uselessly complex mixtures, asphalts. Theory that enumerates small molecule space, as well as structure theory in chemistry, can be construed to regard this devolution a necessary consequence of theory. Conversely, the literature reports, to our knowledge,
exactly 0 confirmed observations where evolution emerged spontaneously from a devolving chemical system. Further, chemical theories, including the second law of thermodynamics, bonding theory that describes the space accessible to sets of atoms, and structure theory requiring that replication systems occupy only tiny fractions of that space, suggest that it is impossible for any non-living chemical system to escape devolution to enter into the Darwinian world of the living. Such statements of impossibility apply even to macromolecules not assumed to be necessary for evolution.
Okay, on to Jim's fourth prompt, which is entirely pseudoscience. Specified information doesn't mean anything. It was coined by Bill Dembski of the Discovery Institute, and it is creationist propaganda. Google the term, and the first hit will be specified complexity, which is described as a creationist argument introduced by William Dembski to promote the pseudoscience of intelligent design. Any code needs a mind to create that code, the usual bullshit. The DI clearly coached him on this, so let's watch it backfire in his face.
Specified information. We have to have some information for the coding in a cell. Here is Lee Cronin, your favorite counselor on Origin of Life. Here's what he says. He says, because there's some contingent information embodied outside the genome. He goes through and he says how people have claimed to have made cells. This is where he's explaining what he means by origin of life research is a scam. It is a scam. I say they're clueless. He says scam.
As you can see, he is now describing synthetic biology, which somewhat overlaps with, but is not actually part of origin of life research. And then he continues to quote mine Lee Cronin. At any rate, I immediately cut through his bullshit and told him that the sequence of nucleotides in a nucleic acid is the information. That's it.
In our view, an explanation of life's origin is fundamentally incomplete in the absence of an account of how the unique causal role played by information in living systems first emerged. In other words, we need to explain the origin of both the hardware and software aspects of life, or the job is only half finished. Explaining the chemical substrate of life and claiming it as a solution to life's origin is like pointing to silicon and copper as an explanation for the goings-on inside a computer.
And decides to write clueless next to polysaccharides before we even discuss them.
You don't have enzymes because you couldn't make them here. You couldn't make them there. No enzymes. You're lost. We already established you don't have enzymes. There's no enzymes yet on the prebiotic earth. You couldn't make them. You're clueless on that.
Okay, time is up.
All of this because he knows full well that the answer to this question is enzymes. One word, and his entire prompt is rendered meaningless. As I've stated multiple times in my content, when James complains about this topic, he is ignoring the very reasonable proposal that enzymes can have come about to then produce biologically relevant disaccharides and polysaccharides prior to the emergence of life, just like other enzymes do it today. These enzymes could be nucleic acids, they could be proteins, but James ignores this obvious solution and just tries to keep jumping back to the idea that we can't get those molecules prebiotically, even though we just spent about 20 minutes talking about how we totally can. I show how childish this is and press him on the idea that given nucleic acids and peptides, getting polysaccharides is not a problem.
Will you concede that this is a perfectly viable explanation for biologically relevant polysaccharides? Enzymes did it, just like they do today. Can you wrap your head around that, James?
Give me a minute to wrap my head around it.
Give you a minute to know what enzymes are and what they do?
Just give me
If enzymes exist, can we get biologically relevant polysaccharides? Yes or no?
James can't even admit that because it invalidates his entire talking point. Then, instead of formally conceding this, he covertly concedes it by then trying to complain about the enzyme that does this chemistry today, and implying that only that enzyme could ever have done this type of chemistry, which is idiotic.
Obtaining prebiotic monosaccharides and oligosaccharides is challenging due to the complex synthesis under prebiotic conditions, the requirement for selectivity in stereoselectivity, stability and reactivity issues, and difficulties in analytical detection and characterization. The challenges associated with the formation of prebiotic monosaccharides and oligosaccharides require precise control over reaction conditions, stereochemistry, and molecular interactions, suggesting a high level of information and intelligence involved. An unguided and random process on a prebiotic earth is highly unlikely to account for their origin. Instead, an intelligent agent provides a better explanation for the precise design and formation of these molecules.
Shows replicators that evolve by natural selection, because it does.
No.
Do you?
No.
Oh, okay. So Gerald Joyce is a fraud. You're calling Gerald Joyce a fraud right now? You should be careful right now.
No, because he doesn't.
No, the paper doesn't do what it says it does. Then, I missed this during the debate, but he starts talking about DNA instead of RNA, which is hilarious.
Copy DNA. How much of the DNA did he copy? You read the paper, I read the...
Fully self-replicating. Fully self-replicating.
No, he did not fully self-replicate it. There's not more than 10... Show me. You got the paper there. No, show it.
I'll save you the agony of reliving this entire exchange, but it's just James insisting over and over that none of these ribozymes fully self-replicated, even though they all describe exponential amplification, which is totally impossible and meaningless if they were not fully self-replicating.
It is not fully replicated. If it was fully replicated, he would have said fully replicated. It is not.
20-fold amplification.
They take a long bit and they duplicate this much of it.
James, how would it retain catalytic activity?
Even if they could duplicate the whole thing, you're
James
still
is
stuck
baselessly denying a body of work and pretending that no one will notice and pretending that and hoping you'll just believe him.
In 2012, 3 years later than the paper cited in the debate, from 2009, Joyce wrote, "In what has been referred to as the molecular biologist's dream, one might imagine that all of the components of RNA were available in some prebiotic pool, and that these components could have assembled into replicating, evolving polynucleotides without the prior existence of any evolved macromolecules. However, a thorough consideration of this RNA first view of the origin of life inevitably triggers the prebiotic chemist's nightmare, with visions of the intractable mixtures that are obtained in experiments designed to simulate the chemistry of the primitive Earth. Perhaps this continuing nightmare will eventually have a happy ending. However, the problem of the origin of the RNA world is far from being solved, and it is fruitful to consider the alternative possibility that RNA was preceded by some other replicating, evolving molecule, just as DNA and proteins were preceded by RNA.
Clearly, James acted like an unhinged maniac, which made good faith discussion impossible, so
James is a chemist and also an apologist who lies about origin of life research on the internet. He sometimes pretends to leave room for it, as he did just now, to seem open-minded, but he isn't. He's totally dogmatic. Therefore, his opinions on origin of life research are irrelevant, as he is approaching the field not as a scientist, but as a preacher. Of course, James pretends his rhetoric is scientific. He will bring up his H-index and list of publications. Less important is the fact that he has publicly admitted that his students do all the work and he slaps his name on the papers. More important is the fact that none of his research has anything to do with origin of life whatsoever and does not qualify him in this topic, which is not strictly synthetic organic chemistry as he claims, but also astronomy, geology, physics, and lots of other areas James is clueless about. When it comes to this topic, James is a YouTuber. His embarrassing commentary takes place exclusively on YouTube or in-person events for science-illiterate Christians who share his biases and delusions. James knows that science is done through the primary scientific literature, and he knows that his inability to publish anything on this topic makes him completely irrelevant. That's why he regularly lies and pretends that he has published in this area, and his papers are ignored because of conspiracy. These papers are actually blog posts in Inference Review, launched by Discovery Institute propagandist David Berlinski, which is not peer-reviewed and exists for the sole purpose of
Right now, today, as a candidate, he supports federal taxpayer funding for Planned Parenthood. I disagree with him on that. That's a matter of principle, and I'll tell you.
You are the single biggest liar. You probably are worse than Jeb Bush. You are the single biggest liar.
All right.
This guy lied. Let me just tell you, this guy lied about Ben Carson when he took votes away from Ben Carson in Iowa, and he just continues. This guy will say anything. Nasty guy. Now I know why he doesn't have one endorsement from any of his colleagues.
All right, John, I get to respond.
Pick from the buffet there.
He's a nasty.